Hello everyone. So in this video, we're going to be looking at the development of new seed. How do seed develop? This particular topic is very important for Waec, Jamb, and Neco candidates. So pay close attention. So it all begins by the differentiation and development of what we call the pollen grains. Now you know that pollination has to do with when the pollen grains, mature ones, are transferred from the anther, which is part of the filamentous part of the plant, to the stigma of the same or another plant. Then that begins the whole process of fertilization. So let's look at how it happens. It all begins with the process of microsporogenesis. So that is what we are going to be talking about here, microsporogenesis. That is the development of the pollen grains. Now, the pollen grains has different parts. We are going to first look at the different parts of the pollen grain before we look at the developmental stages. So the pollen grain is made up of an inner and outer layer. Okay, so this outer layer here, right, is called the exine. The exine, the outer layer that protects the nucleus or the internal part of the pollen grain. Then we have the second layer. So when you come down to the second layer, you have what we call the intine. So the internal layer. So we have the exine, you have the intine, before you have the cytoplasm and other part. Also, there are two basic nucleus in the pollen grain. The pollen grain is made up of the first type of nucleus, which we call the pollen tube nucleus. So this is called the pollen tube nucleus. Pollen tube nucleus. And this is called the generative nucleus. Generative nucleus. All right, so let's begin the process of development. So after some time, this pollen grain will begin to germinate. Okay, so we are going to demonstrate the germination of the pollen grains with this other diagram. So let's assume that the pollen grain lands on the stigma, because when it lands on the stigma, it begins to germinate down the style. So when it lands on the stigma, the first thing that happens is that the pollen tube nucleus remains the way it is. So we'll have the pollen tube nucleus over here. So this is the pollen tube nucleus. It remains like that. However, the generative nucleus will divide into two male nuclei. So instead of having one generative nucleus, you would have the first generative nucleus and the second one. So you now have two male nuclei, this and this. Then you have the pollen tube nucleus. So the next thing that happens is that this outer part, the exine, which is the outer part of the pollen grain, begins to lyse, it begins to rupture. The second layer also begins to rupture. Then this part, which is the intine, right, begins to germinate out. So the intine will begin to germinate out just like this as a result of the rupturing of that layer, the outer layer. So it begins to germinate. So you now start seeing something like this. So in case you are asked to draw the germinating pollen grain, this is what you should draw. But if you are asked to draw a normal pollen grain, this is what you should draw. So the moment this germination begins, the first thing that happens is that the pollen tube nucleus leads the movement. Okay, after the pollen tube nucleus starts the movement, the generative nucleus as well follows the pollen tube nucleus. Generative nucleus follows the pollen tube nucleus. So you have the three nucleus going down, germinating alongside the pollen grain. Now, the major function of the pollen tube nucleus, the major function of the pollen tube nucleus is the major function is direction of the pollen tube. So the pollen tube nucleus directs the pollen tube. At some point, the pollen tube nucleus will disintegrate or will degenerate. Okay, now this happens when the pollen grain lands on the stigma. So you have the pollen grain here germinating, trying to germinate down the style. So you have the first nucleus, which is the pollen tube nucleus, and you have the next two that follows, which is the generative nucleus. The nuclei now, the two male nuclei, because they've differentiated from generative nucleus to the nuclei, two male nuclei. All right, so let's take a magnified part of this structure. What happens inside the ovary? Okay, so it's like now we have gotten into the ovary. We have gotten into the ovary, and there are different ovules of the ovary, right? So what we are doing is that we are taking just one part and magnifying it to this other structure here. Okay, so now let's look at what happens here. When the pollen tube nucleus lands on the stigma, it grows down. So let's draw the growth. So let's assume this is the pollen tube nucleus growing down the style. Now it comes this way. Remember that it is being led by the pollen tube nucleus. Okay, so you first have, at first, you have the pollen tube nucleus. Then you have the male nuclei following. So this continues until it gets to this point. Now, before we go into the whole process, let's look at the different parts of the ovule. Now you can see that there are some cells here. These cells above that is connected, that is attached to the margin, to the upper margin of the ovule, is called the antipodal cells. It's called the antipodal cells. And what is the function of the antipodal cell? It is actually a nutritive cell. It plays a nutritive function. So it gives nutrient to the ovule. So let's move to the next one. This central cell is called the central cell. The central cell. Or you can call it binucleated cell. Binucleated cell. Because it has two nucleus. All right. The next one is this particular cell here. So you can see that the cell is being hugged by two other cells. These two here, this one by this side and this side, they are called the synergid cells. They are called the synergid cells. Synergid cells. Synergid cell. Now we also have this one, which is one of the most important cells in the ovule. It's called the egg. The egg cell. So these are the different parts of the ovule we have. All right, so let's go back to our germination process. So this begins to go down. So, okay, we have another part, which is the opening. It's like a little pore that lets the nuclei into the pollen grain, into the aperture of the pollen grain, of the ovule rather, into the aperture of the ovule. So let's label that. So this part is called the micropyle. Micropyle. So when this gets to the micropyle, this is what happens. The moment this part touches here, the micropyle, the moment it touches there, this wall of the ovule disintegrates. Okay, and that is a function of the pollen tube nucleus. The moment the pollen tube nucleus gets to this part and it touches this membrane of the ovule, that part disintegrates, leaving a space. So it disintegrates with the pollen tube nucleus. So the pollen tube nucleus has finished its course. So what is left are the two nuclei. So you have the two nuclei trying to gain entrance into the aperture of the ovule. Now this is where the whole idea of double fertilization comes in. The first nuclei will fertilize, will fuse to the egg cell. So this first nuclei will enter into this point. It can pass through, you can pass between the synergy cells, or you can go this way and fertilize and fuse together with the egg. Now, when this fusion occurs, the product that will be formed is called the zygote. And the zygote will later develop or differentiate to form what we call the embryo. The embryo. All right. So the next fertilization that occurs, remember, this one has gone, the first one has gone. So we are left with just one. So this one also comes in and goes to the binucleated cell at the middle of the ovule. Now, when it gets there, it fertilizes, it fuses with this other one. Okay. Now, this structure is called the triploid. Triploid, because for this particular one, it is diploid, because you have the first, the male nucleus fusing with that of the female. So we have the male having N, the female having N, so making it 2N. However, in this one, we have the one from the male, which is N, then two other, because it's a binucleated cell. So it has two nucleus. So N here, N here. So we now have 3N. So that makes it a triploid cell. So this is the idea of double fertilization. The first fertilization happens when the first nucleus fertilizes the egg, giving rise to the zygote, which develops to the embryo. The second nucleus moves to the central cell or the binucleated cell and fertilize the cell, giving us a product which is a triploid cell, and it is 3N. Now remember I said that the zygote develops to form the embryo. Meanwhile, for the triploid cell, it actually develops to form what we call the endosperm. The endosperm. Or you can say the primary endosperm. The primary endosperm. And that is what forms the outer part of the fruit. Meanwhile, the embryo actually forms the seed, right? The embryo in the seed. That is what this particular product forms. Then the triploid cell forms the endosperm. So this is the whole process of double fertilization in plants. So I'll do a very quick run through. We have microsporogenesis, which has to do with the development of the pollen grain, right? From this part, from this structure into this structure, which is germinating. So we assume that this structure falls on the stigma. Okay, so when pollination occurs, the pollen grain falls on the stigma and begins to grow down the style. Now we now said we picked one of the ovule and magnified it, and that's the structure we had here. So when the pollen grain falls, it begins to grow. The first nucleus that leads the way, that directs the pollen grain, the pollen tube nucleus rather, is called the pollen tube nucleus. The second one that follows are the nuclei, which are like daughter cells of the generative nucleus. So they follow. The moment the pollen tube nucleus touches these segments of the ovule, it disintegrates, leaving an opening for the two nuclei that followed it. So when it enters, the first one will fertilize this egg cell, giving us a zygote, which is 2M. The second one will go and fertilize the binucleated cell in the middle of the cell, and that will give us a triploid cell, which form the primary endosperm. We also have, I talked about the antipodal cell. This plays a nutritive function. Okay, so this is the whole um, process of development of new seeds.